Hello to everyone and welcome to the Modeling Speaker series. Today we have a very special guest, Guillaume Fournie. He's a veterinarian and epidemiologist at INRAE, the French National Research Institute for Agriculture, Food and the Environment. He also holds an honorary appointment at the Royal Veterinary College in London. His research explores how livestock production systems shape the emergence and spread of animal and zoonotic pathogens, in particular focused on the role of intensified poultry production and the transmission of avian influenza. He has increasingly been working at the intersection of epidemiology, virology, and social sciences, and yet we are very thrilled to have you here today, Guillaume. Just a quick note before we begin. Today's session is being recorded and may be shared with the board. So if you have any concerns, don't hesitate to reach out to us. And with that, I will hand over to Guillaume for his talk. He'll take questions at the end of the talk. You can either raise your hand in WebEx or drop them in the chat. We will read them out loud. So over to you, Guillaume. Take it away whenever you're ready.
Okay, thank you very much, Jose, for the invitation. Today I will talk about some work that we conducted on the transmission and control of avian influenza viruses along live poultry trading networks. I will talk about models, but not only models. I will also talk about other research which was used to that we used to inform the development of the models, so parameterization, and also to interpret the results. So I will also talk about some fieldwork we did, biological fieldwork, but also some social science fieldwork that we did as part of the UKRI GCRF One Health Poultry Hub, which is a large-scale project, an international project that involves about 27 partner institutions, and which focus primarily on the intensification of poultry production and its consequences for health in Southeast Asia, in South Asia and Southeast Asia, in 4 countries mainly: India, Bangladesh, Sri Lanka, and Vietnam. Most of the work I will present today is focused, as I said, on avian influenza and on Bangladesh. So the work I'm presenting is not mine. It has been done by a number of people. For sample collection, most of the work I will present are based on data collected by Natalie, Shuman, Rashid, Lisa, Hassan, and Matt. The phylodynamic and genetic analyses have been undertaken by Jaina, Lorcan, and Rebecca, and the statistical and mathematical modeling by Marie-Cécile and Francesco. So just a bit of context. Poultry production is now the main livestock sector globally. You see here the evolution of the production of poultry meat over the year, and you see that it's in blue for poultry and in a dotted line for chickens, and you see that the production increased by about 15-fold in only 16 years. And this increase in production is heterogeneous spatially, with Asia accounting for most of this increase in production over time, and within Asia, South Asia, sorry, in which the production in poultry meat increased more than 60-fold in 60 years. And when we think about poultry intensification, we think mainly about the image on the top left here of the slide. This big shed full of white feathered, phenotypically identical chickens. But actually, in low and middle income countries, which are intensifying the production now, there are lots of different modes of production and then of commercialization and of slaughter and marketing of those chickens, especially with live bird markets often playing an important role in the trade of those chickens and playing an important role in the food security in those countries. The importance of live bird markets in the epidemiology of avian influenza viruses has been known for decades, but became really, started to attract a lot of attention in 97 with the first outbreak of H5N1 in Hong Kong, which caused the death of a few people. When outbreak investigations were made, chickens which are highly susceptible to H5N1 and die within a few hours or a couple of days, these chickens were found, the virus was found to be highly prevalent in chickens offered for sale in the markets of these cities. About 20% of the chickens were found positive despite not showing any clinical signs, which was surprising. The hypothesis that was developed at the time was that this chicken may have had some level of cross-immunity with other, induced by other circulating avian influenza subtypes, less pathogenic. Another possible explanation was that the live bird market created an environment which was promoting the amplification of the viral circulation and its maintenance, just due to the way the market was functioning. And this is what we investigated here with a mathematical model, which basically is a simple mathematical model which takes into account the population dynamics of chickens in the market. So when the market is open, it will receive batch of chickens at the beginning of the day, then chicken will be sold, and chicken left as surplus will be stored during the night and reoffered for sale the following day when more susceptible chickens will enter the market. And so you see here the result of this mathematical model with the y-axis, the prevalence as a function of time during opening time, night when the market is closed, when it's open again, and so on. So you see here in blue the prevalence of infectious chicken, and you can see that we can have a very high level of infectious chickens being offered for sale in the market, while the prevalence of chickens showing clinical signs remain fairly low. And this is because chickens will basically remain enough time in the market to become infected and shed virus and contaminate other, but will be slaughtered before showing clinical signs of dying of the disease. So this type of markets may create conditions for the silent amplification and maintenance of these viruses acting as reservoirs. So what we wanted to see is now in countries where the viruses are endemic, which is not the case in Hong Kong, where this epidemic and subsequent one were rapidly controlled by culling, but in countries in Asia where this virus is endemic in the poultry population, whether we see similar patterns in the poultry production systems, in the marketing systems, and what are the characteristics that promote this maintenance of the virus. So we focused on Bangladesh and whether multiple subtypes of avian influenza are endemic, and we focused on two main subtypes, H5N1, which is highly pathogenic, and H9N2, which is highly prevalent in chickens, which is low pathogenic, but nonetheless causing a substantial drop in production, which is a major concern for the sector, but also being a major gene donor in the emergence of other variants, and being itself a zoonotic variant as well. So we started by characterizing the system. And by doing that, we started by identifying the types of roles and the type of actors involved in the production and distribution of chickens. And we went beyond the description of the flow of commodities, so just the flow of chickens, by characterizing how the dynamics of these roles affects and is affected by the structure within which actors operate. So in Bangladesh, the transaction dynamics was a complex mix of unpaid domestic labor, patronage, and various forms of value appropriation. The lack of access to financial capital meant that many farmers had to rely on credit from suppliers to gain access to production inputs, such as day-old chicks, feed, medicine, placing them in interlocked transactions, which were characterized by price fixing and trade-tying contracts. There was also a multiplication of roles with many opportunities for actors to occupy network niches in order to earn an income. And there was also the incessant background pressure from integrators seeking to appropriate value at all points from hatchery to final consumer. So this all meant that most of the production occurred within fragile enterprises, with individual agents making decisions on the basis of short-term goals rather than long-term consideration, adopting reactive rather than preventive and proactive measures. and with this decision having possible implication for viral transmission. So based on these qualitative insights, we then quantified the movement of poultry from producing to marketing areas. We started in the market and then traced back the origin of the poultry. These allow us to identify the supplying areas of markets. So you see here for two different cities, Dhaka in the center and Chattogram in the south east, which are the two main urban centers in the country, and for different types of chickens. And then we use this information to develop gravity models, which are statistical models used to model flow of commodities or flow of people, in our case, flow of poultry, of different type of poultry, using as predictors the density of poultry and the proximity between supplying and selling areas. And so we could then apply this gravity model, fit it for the data we have to cities for which we didn't have data. We also characterize the movement of traders between markets within cities and the poultry population dynamics for different types of poultry within the markets in the different cities. So this allowed us to basically characterize how the trade of poultry could connect epidemiologically regions that would have otherwise remained isolated. Then, based on this characterization of the network, we then conducted biological studies to identify, to quantify the amplification of the viral circulation. So in Bangladesh, like in most countries where these viruses are endemic, what we find is a spectrum of amplification from farms where whatever the subtype, of course, the prevalence will differ according to the subtype, but for any subtype, we'll find very low to absence or near absence of avian influenza at the farm level, but very high prevalence in markets. And here I'm just changing a bit. We did also some work in Vietnam. So just to mention some work in Vietnam, when we look at the prevalence in markets where, like in Bangladesh, these viruses will be ubiquitous and present usually at high prevalence. What we observe is that the prevalence in markets is associated with practices in the markets, such as increased length of stay, so the length that chickens spend in the market, but also associated with supply features, such as the length of the supplying chains, the number of actors involved, and the location of farming areas supplying those markets, rather than the actual location of the markets. So the shedding of virus in markets, and more generally in the endpoints, whether they are slaughterhouses or markets, may not only be affected by the viral transmission in the sites, but also by transmission events occurring upstream along the network. So what we wanted to do is to quantify the contribution of these different transmission routes to the shedding of viruses in markets. And so to do so, to quantify this, we conducted a field experiment. And through this field experiment, we basically altered the trading network and repeatedly sampled chickens for a period of time at the farm gate, and then at the market gate, and then different time during a few days. And these data were then used to quantify the increase in prevalence during the stay in the market. And as we see here, by 12 hours, while we didn't see much difference in the prevalence at the market gate between chickens that transited through our altered system and chickens that transited through the conventional market chain, by 12 hours, we find a quite an increase in prevalence in the conventional system. So meaning that viral transmission, which happened before the market, played an important role in what we see in the markets. So we then use this data to fit a mechanistic model, which simulated the transmission of H9N2 in a market, accounting for the population dynamics of the chicken in the market. So we consider direct and indirect transmission, and we fitted this model using MCMC approaches. And this allowed us to estimate different parameters. So here for the parameters, you see different curves which are related to different criteria for classifying a chicken as positive based on the t-value. For high CT, why we did that is because to reduce the likelihood of considering a chicken positive when it could have only been contaminated with RNA virus, with RNA being present in the air and in the nostril of the chickens. So you see here at the bottom left, you see the distribution of R naught in the market. So we have a relatively high R naught in those markets, and this is associated with very short latent period. So the period from onset of infection to onset of infectiousness. And this is basically about 6 hours for broilers. Broilers as this white feathered exotic breed, which represent most of the chickens sold in this market, and a bit longer, but still relatively short, around 1 day for backyard chickens, which are indigenous breeds of chicken, often hybrid. So the combination of short infectious period and high level of transmission with high R naught meant that chickens entering the market as susceptible quickly become infected as they spend time in the market. So most of the chicken entering the market will ultimately be exposed to the virus and become infected. So there is a high level of transmission within those markets. But what we see as well is that there is a high proportion of chickens entering the market as being already infected. So you see here the sum of the pre-infectious and the infectious chickens here in broilers, which again account for most of the chickens being sold in those markets. And about 10% of them enter the market as being already infected. Backyard chicken, which represents a small fraction, a much smaller fraction, this proportion is lower, but what you see as well is that there is a higher proportion of chickens that may already be immune, so have been exposed before entering into the market. And this is because these backyard chickens have a much longer life expectancy than the broiler chickens. Broiler chickens are raised for about 30 days, when backyard chickens are raised from 6 months to 2 years. So what we have here is that for H9N2, we have a high level of viral amplification in the market, but the level of shedding that we observe is also driven by transmission which occur upstream during transport, but also potentially in the farms. And indeed, when I mentioned earlier that when we go and conduct cross-sectional studies, so sampling chickens during the production cycles on farms, we barely see any, we barely detect any virus, very rarely. But when we did some additional work where we purposively sampled chickens at the very end of the production cycle, just before chickens were starting being traded, and then at the very end when once the batch was emptied. A big difference with Europe, for instance, is that in Bangladesh, like in Vietnam and many countries, in many countries in Asia, all chickens are not sold in one go to the market, but are sold over a period of time, which can take a couple to 10 days to sell a batch of chickens, with multiple traders coming during this period to collect birds. And this is a response to the volatility of the market, with price fluctuating a lot. So this is a response of the farmers to the socioeconomic context in which they are operating, in order to basically reduce the impact of these price variations on their income. And so this is also identified as we also identified this as a high-risk period for the introduction of the virus on those farms. But what about H5N1? So for H5N1, we have a very different picture. Chickens represent about 99% of the poultry being sold in those markets, and ducks about 1% or even less, let's say between 1 and 2%. And they will not be sold in all the markets. And when we conduct virological studies, we find that the prevalence of H9 and H5N1 in chickens is about 1%, when in ducks is much, much higher. So this suggests that ducks play the main role here in the introduction of the virus in markets and in its amplification in markets. And indeed, we at the moment conduct a small pilot study in Bangladesh, where we sample ducks, similarly a simplified version of the experimental study I mentioned earlier, where we sample ducks as they are delivered by traders and then for a period of time in the market, and we find pretty high prevalence already among the chickens out of the truck. So suggesting that indeed ducks play an important play an important role in the introduction of the virus in the market. And as well, we find higher prevalence of chickens which are in contact with ducks than in chickens which are not in contact with any ducks, which are in stalls where ducks are not offered for sale. So this really suggests that ducks is driving the dynamics of H5N1 in the markets. So the non-detection of H5N1 or the very low level of detection of H5N1 in chickens, especially in broilers, the fact that we don't detect H5N1 or very rarely during cross-sectional studies, and this combined with the very low reporting of outbreaks on farms in Bangladesh, all this, and then when we consider as well the high prevalence of the infection in marketing ducks, resulted in the epidemiological narrative that chickens production did not contribute much in the maintenance of the virus, that most of this maintenance was due to ducks and backyard chickens, but not with commercial chickens. So we wanted to explore this a bit further. And we first investigated farms experiencing unusual mortality and morbidity outbreaks by sampling chickens if farmers sought veterinary support. And so we did two different studies, and through the two studies, we found that a high proportion, about 10% of the samples of those farms, in these two successive cross-sectional studies, were being positive for H5N1. So outbreak of H5N1 remain common in commercial farms, even if they are not reported, and even if incidents due to the nature of the survey population here is very difficult to quantify, very difficult to understand what our denominator is. But what we know is that it's not infrequent to find H5N1 in farms. So what we did after is that we conducted a longitudinal study on broiler farms where we followed 100 farms, so it's still a fairly low number, but 100 farms, but from start to finish, and we sampled at several time points. And here we found that 6% of farms had been positive for H5N1 by the end of the production cycle, mainly towards the end of the production cycle. We are now conducting this study at a larger scale, but all these suggest that the infection is much more common than what we thought it was in chicken farms. And this is because H5N1 infected chickens. Why do we don't find H5N1 in chickens in the markets? Because those H5N1 infected chickens may not end up in markets, but be traded through alternative channels. So they qualify as a commodity, but as a different type of commodities and healthy appearing chickens. In Vietnam, we had evidence for very distinct traders that were specialized for specific qualities, so-called qualities of chickens, and in particular for dead or very sick chickens indeed. But so the fact that you have outbreaks on farms, even lots of outbreaks, doesn't necessarily mean that commercial farms play a role in the transmission of the virus. You could consider them as a dead end, with farms becoming infected, but not necessarily transmitting the infection. They could be just spillover from local markets. So what we did is that we then sequenced the viruses we found on farms. So you see here a molecular clock, a time-scaled phylogenetic tree. With each tip, non-marked tips are sequences found in markets, mainly ducks, and in markets in Dhaka and Chattogram. And the circles are sequences from found on farms that I mentioned in Chattogram and Dhaka, in the supplying area, belonging to the supplying area of those markets. And what you see is that you have farms which form their own monophyletic clades. So meaning that you have a monophyletic group. So you have this sample from farm clustered together. So meaning that there seems to, and away from, for most of them, away from the tips we find in markets. So meaning that there is possibly some chains of transmission between farms. And so therefore, these commercial chicken farms may play a role in the spread of this pathogen. I'm not undermining the importance of ducks in the epidemiology of H5N1, but just highlighting that we may have underestimated the importance of chicken farm. More importantly, because those chicken farms are not vaccinated, the broiler farms are not vaccinated against, these broiler chickens are not vaccinated against H5N1. The parents are, but not the actual offsprings, making them susceptible, and with the addition of maternal immunity, potentially even promoting the late infection of the chicken during the production cycle, and allowing, because the late infection is late, allowing farmers to sell those chickens because they will be detected as infected as a fairly low stage. And this is something that we're now investigating through a mathematical model, whether we can identify high prevalence level of infection in those chickens, despite having a resilient system that can still produce poultry meat for the population. So now we based on this characterization of the network of production and distribution of chicken and this viral amplification of the virus along the trading network, we use that to make sense of the genetic data we generated. And so first we looked at a situation within markets. There we found very high level of, for H9N2, very high level of lineage turnover. So here is for 2 markets where Lorcan sampled every 2 days over a month. And what you see that each line is a lineage and each dot is a tip, is an occurrence, a sequence. And we find lots of different lineages circulating at any one time in the market. But quite importantly, while some lineage can circulate in the virus in the market for several days, lots of lineages are only found 1, 2 or just over a defined period of time in those markets. So this allowed us to, according to different assumptions, I will not go in details about that because of time, but what we find is that therefore there is a limited duration of circulation of those lineages in those markets. And what we're now looking at is with a multi-strain model, which is fitted based on structured coalescence model. We are estimating the mean extinction time of lineages in those markets. So you see here for different values of R0 in the market, we estimate the mean extinction time, which is basically the duration during which a virus which is introduced in the market will be found circulating in that market. So that's the mean. I'm not showing here the overall distribution, which is really right skewed. But what you can see here that this mean extinction time will increase with R0 and will also increase as the color of the slide become darker. And this is linked to the proportion of poultry being left as surplus from one day to the next. Because in this market, because of the high level of viral amplification, a large proportion of the surplus poultry will actually be infected, and then will allow the introduction of the virus, the introduction of the recirculation of the virus from one day to the next. So you see that there is a balance between the level of surplus that you have in those markets, but also with the x-axis, which indicate the prevalence of the infection at entrance in the market. So if the prevalence is high, the lineage, the length of time that the lineages circulate is low, as you have a new introduction of new lineages every day that compete with the existing ones. But if the level of surplus is high, then the level of extinction that those lineages will circulate for a longer period of time because they will be represented by a larger number of chickens when the new chickens enter the market through this surplus. We then looked at the spread between markets, and this was done by sampling chicken through cross-sectional studies in different markets in different cities across the country, and conducting phylogeographic analysis. And what we find is that the supplying areas, which is again the spatial distribution of farms supplying a market, was a strong predictor of viral dispersion, which meant that cities with a high number of supplying farms in common, so 2 cities with several supplying farms in common, have greater virus movements between them. So interestingly, we also found that this was significant for different chicken types. Now, within cities, we found a very high level of diversity intra-market compared to inter-market. So meaning that within a city, each market accounted for a very large proportion of the genetic diversity that you could see at the scale of the city. And the configuration of the trading networks in those cities strongly influence H9N2 dispersion between the markets in the city. So indeed, trading practices along this network promoted a mixing of chickens. And any two chickens supplied to a city could potentially be traded in the same market, regardless of their origin, as long as it was within, of course, the supplying area. And therefore, these practices promoted also viral mixing, resulting in a high viral genetic diversity in any market. And these patterns have a direct implication for the design of surveillance programs. Indeed, to improve cost effectiveness, instead of, as done now, where a large number of markets are targeted in the capital city, it will be much more effective for early detection of new variants to target a small number of markets located in cities with non-overlapping catchment areas. And this can be predicted with the gravity model I mentioned earlier. That was for H9N2. For H5N1, we adopted a different sampling strategy as we wanted to monitor the introduction of 2344B's new clade, which is causing the current global spread. And we also focused on areas where we had different level of duck density. And what we found is, on one hand, for 2344B, we found several introduction of the virus, which correspond in the country, which corresponded to the period during which wild birds winter in Bangladesh. And this was linked to wild bird sequences found elsewhere in Asia. And we found a rapid dispersion of the viruses once they were introduced. They were found in lots of different locations across the country. For the clade 2321A, which is a historical clade which has been endemic in Bangladesh for several years, we found that first it was less geographically structured than H9N2, and our hypothesis that we are testing now, similarly as we did for H9N2, that it is linked to the transport distances of ducks, which is much longer than the transport distances of chicken, and which may result in this less structured genetic structure geographically. And we found as well that there is an increase in the relative proportion of sequences which are represented by 2344B compared to 321A. So whether this will lead to a replacement is an open question. For the moment, still in 2024 and early 2025, we still find 2321A. I don't have like new figures about the relative prevalence between these two subtypes to share, these two clades to share now, but whether or not it will lead to replacement is still an open question. And so now what we're doing is that we're taking this phylogenetic data, all these data about the structure of the systems and about the about knowledge about dispersion pattern of the viruses to feed into an individual-based mathematical model, which is EpiNest. It's a modeling framework that we developed, primarily Francesco, who developed it, which allowed to model both synthetic networks, so basically to reproduce in silico networks of trade from farms to end users, and also to simulate the spread of pathogen through these networks, and to consider different epidemiological scenarios about where the amplification of the virus may happen in farms, in markets, in middleman stage, or at different stages, therefore, along the trading network, and allow us as well to account for the spread of multiple strains, as well as to reconstruct, because the individual-based model, to reconstruct transmission trees based on the model, and to compare these transmission trees to phylogenetic trees that were inferred from empirical data. So our ambition here is basically to calibrate those models with the phylogenetic data we generated. It's something that Francesco is now working on. So can I take just 4 minutes to talk about risk mitigation, Jose?
Sure. Yep. Go on.
Okay, thank you. So in terms of risk mitigation, the often when markets are associated with infection, especially zoonotic infection, the first thing which is being done is to ban the market and to close them. This strategy has been shown to be quite effective, especially in China in around the outbreak of H7N9 10 years ago, but very difficult to sustain the long term, even over there. So an alternative approach which has been developed in Hong Kong is a set of technical interventions which are disinfections and reduction of the length of stay of poultry in the markets. So different interventions which aim to reduce the level of viral amplification in the market while minimizing disturbance for the poultry traders. As you can see here, this is the prevalence of H9N2 in markets in Hong Kong, and it decreased over time when different strategies are implemented, and especially one which was very effective was the ban on overnight poultry storage. So with the model I mentioned earlier of H9N2 spread in markets, we replicated this type of intervention in our in silico setting. So what you see here is a prevalence reduction, so the level of prevalence reduction according to the level of disinfection. So a level of disinfection of 0.4 means that every day we remove 40% of the virus from the environment. Don't pay attention to the different colors or the different texture of line, just look at the overall trend. So what you see that if only disinfection is implemented, partially effective intervention, but it's implemented every day, you have, of course, a reduction in the prevalence, but which is not effective enough. When it is associated with what I mentioned earlier, a ban on overnight stay, so basically the stay cannot be more than a day, then combined with disinfection, it reduces, again, further the prevalence, but to an asymptote. So you have an impact, but which remains limited. Here in our setting, if you really want to have a strong impact and to reduce drastically the prevalence, what you need is to also reduce the prevalence at entry in the market. And this is a challenge we have in those settings. So the Hong Kong experience has been very instrumental in the way strategies wasn't developed for the rest of Asia and also Africa, with a focus on markets and strategies implemented in markets. But what this model is suggesting is that these strategies, even if they were perfectly implemented, may only have a limited impact, because in those settings where you have a very high frequency of introduction of the virus into those markets, targeting markets alone is insufficient to achieve a reduction in viral shedding in those markets. You need interventions along the entire network, which of course is extremely complex. It's even more complex because these are technical interventions, and therefore they often ignore the context within which the trader operates. Traders adopt the daily practices for all kinds of reasons, and unless we address them, then we will be very limited in the effectiveness of our technical interventions, even if they were really adapted to the epidemiological context we are targeting, which is not the case now. But this is starting to be recognized now by WHO and in particular in this document. But even if the guidance on how to do that is still missing. I would just finish by saying that we are also at a macroscopic level, we're also very interesting to look into what intensification, the future of intensification means in these settings for the vulnerability of the system as the level of production keeps increasing. And especially we developed a new kind of spatial distribution models in Marie-Cécile who did this work, which is basically account for this, not only the spatial predictors of farm location and farm size, but also of clustering of farms, because in agricultural production, farms are clustered, and this has an important impact on the potential of pathogen to spread, because they will spread pathogens, especially avian influenza viruses, will spread much more strongly in farms, in settings where farms are already clustered. So we adopted a framework, a statistical modeling framework to do that, and we're now with Pauline, a student of Marie-Cécile, looking at how different scenarios of intensification resulting in different numbers of farms, but also different level of clustering and aggregation of those farms may result in different level of vulnerability of those farms for emerging pathogens associated with different characteristics. So you see here the probability of a large outbreak. Each line represents the different scenarios here in the background about clustering of those farms according to different level of transmission for different kernel, which is the long-range kernel, meaning that you allow for the farms to be able to spread the infection pretty large distance, or short-range kernel, which is often more realistic for avian influenza, where the infection can only spread from a farm at short distance. And then you see here that different patterns are associated with very different ability to allow a pathogen to spread in the systems. So in conclusion, we have a high level of viral amplification and mixing, a high level of exposure, therefore, for humans in this market. And this is very much driven by the configuration of production and distribution networks. And this is shaped by structural factors that influence practice and limit the effectiveness of technical intervention, and therefore needs to be targeted if we want this intervention to be effective. And I will stop here. Thank you.
Thank you, Guillaume. Very, very interesting. Certainly a complex problem. Very interesting to see the work that you and your team are doing at various different levels. Yeah, understanding the production systems, the commercialization systems, the epidemiology of disease on top of that, behavioral cultural factors that have an impact, zooming into the virus and the sequences. It's really, really interesting, the approach that you're taking, and likely the only way to really start understanding a bit better, but the best ways to control avian flu spread and risk to humans. So yeah, thank you for that. We now have 6 minutes for questions. So if anyone in the audience would like to ask anything, I have a small question. When you showed one of the, I think it was the first compartmental model, you had an arrow leaving the environment, but then coming back. It was like a loop, and I was wondering what that represented, what you meant by that in that.
Okay, so what we considered is, in that model, we considered 2 transmission routes. So chicken could infect other chickens through direct contact.
Yep.
But also by contaminating the environment of the market.
Yeah.
So therefore, the other loop was basically the force of infection, which was from this environment.
Okay.
So we could not, with our experiment, we could not quantify the importance of environmental transmission versus direct transmission. What we did, and then consider. So what we could do is consider the overall force of infection, whether it's by the cumulated routes, if you want, and then basically varying, exploring the impact of variation in the proportion of the contribution of each route to the force of infection. This allowed us then to account for basically disinfection, because of course if we only consider the direct transmission route, which is fine to estimate R0 and so on in that setting, but it's not enough to it doesn't allow you to assess the impact of disinfection.
Okay, that takes me to more questions on that. For example, thinking how do you account by the decay of the virus in the environment and combine that somehow with the duration of infectiousness in infectious chicken. You have 2 different.
So basically the model could be refitted for different assumptions about this. So it was, I didn't want to already talk too long, so I didn't want to talk to describe things in too much detail, so take too much time. But indeed, if you remember the graph I showed about the effectiveness of interventions, there were several lines, and the several lines correspond to different assumptions about the decay, the survival of the virus in the environment. So this survival of the virus in the environment in our model will have a very limited impact on the fitting of the model, on the adjustment and estimation of R0 and so on, but will have an important impact on the effectiveness of the interventions. But even for very different decays, which corresponded to what we found in the literature, so the variability we found in the literature about decay of those viruses, you still see an overall trend, which is basically targeting only markets will not be enough to achieve a strong reduction in prevalence.
Now, thinking in the broader context of what's happening globally with the spread of avian influenza, and also looking at what's happening in the US with this, what we're seeing with the transmission among dairy cattle. Have you thought about what of how much of your research do you think could help tackle some of the new challenges that we're seeing with increased transmission globally, and in particular to what's happening in the US?
So I mean, what's happening in the US here is not directly relevant. What it is for what we are concerned and wondering, it will be the establishment. So as I was mentioning, there is one main clade which has been endemic for several years in Bangladesh, with 2344B now having established itself in the population for now a couple of years, to see the long-term trend associated to that, and especially whether this pushes towards reassortment of the virus, as we found in Cambodia, where there has been reassortment between the historic clade, which I think 2321C, I think in Cambodia, and 2344B, and which have been associated to human cases, if I'm not wrong. So there is concern there that at least arriving this new this clade, which has been spreading globally in regions where you have a diversity of viruses, that's pretty concerning. What is also concerning is that now, with a change of landscape of aid funding globally, within country like Bangladesh, but the case in most countries in the region, where most of the surveillance was funded based on US funding, that's a major concern. So because it means that very quickly we can all become very blind to what's happening in the systems. So lots of the data. So the data, as I mentioned here, were essentially produced by a research project. But there is additional, there's also make use of other data, which were, for instance, produced by a CEIRR project. So what will be the long-term viability of those projects, which really played a big role in the surveillance of new variants, but also of FAO-funded surveillance, for example, which was directly USAID funded for. In that case, that's a major concern.
Thank you. We are on time already, so if there's any short question from the audience, otherwise we'll wrap it up there. Thank you very much, Guillaume, for your time. Really appreciate you taking the time to come and do this talk.
Thank you, Jose. Nice to see you again.
Cheers. Thank you all, and have a good rest of the evening.